Hi, my name is Jonathan Tran, and I am a student from Harmony School of Advancement. The project that my team and I have decided to present is the science behind gene therapy, more specifically gene therapy with the herpes virus. According to Medline Plus, gene therapy is designed to introduce genetic material into cells to compensate for abnormal genes or to make beneficial proteins. If a mutated gene causes a necessary protein to be faulty or missing, gene therapy may be used to introduce a normal copy of the gene to restore the function of the protein. A gene that is inserted directly into a cell usually does not function. Instead, a carrier called a vector is genetically modified to deliver the gene. Certain viruses are often used as vectors because they can deliver the new gene by infecting the cell. The viruses are modified so they can't cause disease when used in people. Some types of viruses, such as the retrovirus, integrate their genetic material into a chromosome of the human cell. Other viruses, such as the adenovirus, introduce their DNA into the nucleus of the cell, but the DNA is not integrated into a chromosome. The vector can be injected or given intravenously directly into a specific tissue in the body, where it is taken up by individual cells. Alternatively, a patient's cell can be removed or exposed to a vector in a laboratory setting. The cells containing the vectors are then returned to the patient. If the treatment is successful, the new genes delivered by the vector can make the functioning protein. Next portion of our project is the ELA component. Gene therapy is a very modern form of medicine and is quickly propelling the medicine and technological field. This means there is not much prior knowledge of its use and effects. Our ELA component takes the form of a news article in order to be more comprehensive for our audience. It breaks down the skeptical commentary that the public and medical technicians would have with gene therapy and weighs out the pros and cons of its use. This component brings our project back into the real world with genuine questions and concerns. Lastly, our SS component places gene therapy in a more historical situation and social setting, that is, since throughout the course of history, genetic disorders have had an impact on people's lives. As technology and human knowledge and biology have improved, the understanding of genetic disorders have also gotten better. For example, the development of karyotyping has allowed for the mapping of chromosomes, which allows scientists and other biologists to understand more about genetics and how genetic disorders are caused by chromosomal abnormalities. Going into the main topic, single gene mutations have been discovered by using SNAPs and other processes such as PCR and gel electrophoresis. These newer technologies and discoveries have allowed for diagnosing genetic disorders which were not possible to humans before. Thank you for watching Harmony School of Advancement.